Well, Max and Hugo, thank you very much for inviting me to the conference. The foundation that you're launching is going to fill a niche that's very much needed in the philanthropic and funding space. There's plenty of venture capital for seed companies that are preclinical, and very little venture capital for mid-stage companies that have a promising drug that they want to evaluate for the first time in patients. So I commend you for this new model for public-private partnership, and I hope that it's successful. This is the first Alzheimer's Solution Conference. I hope there'll be a number 2 and number 3, and would enjoy coming back at a later date. So we are developing BPN 14770. It's a novel approach to address memory loss in Alzheimer's disease and perhaps improve cognitive resilience, delay progression, or delay onset of the disease. We developed BPN 14770 as a public-private partnership with the NIH through the NIH Blueprint program. We're targeting an enzyme known as PD4D. There are 4 PD4s in the human genome. The very first learning mutations ever discovered in a model organism were 2 mutations in Drosophila, rutabaga and DUNS. And these 2 mutations defined a pathway that's used both in the brains of, brains of fruit fly, and was then the same gene set now is used in mammals. And in mammals, this gene set is put downstream of the NMDA receptor, which through allowing calcium to enter the neuron, activates calcium calmodulin adenylate cyclase, rutabaga, which and in turn that cyclic P signal is ring-fenced in the synapse by PD4. In the case of the human brain, the ring fence is PD4D. So what we've done in our program is develop a selective allosteric inhibitor of PD4D. We just published the chemistry in ACS Journal of Medicinal Chemistry. Our team was the first team to obtain crystal structures of the PD4 enzyme with its regulatory domains. And we were the first to identify a binding site for an allosteric inhibitor. And that gave us a new entry point into drugging this important class of targets. So in addition to our work on PD4D and cognition, we also are very interested in the microglial cells and another enzyme in this family called PD4B, whose activation is important for microglial activation. So this slide shows the allosteric binding site, PD4D on the left, PD4B, which is the counter target on the right. The compound that we developed is the first subtype selective inhibitor. Other compounds have been developed in industry. There are 2 now in the market that are very successful, one of which is very successful for psoriasis and psoriatic arthritis. But we're the first company to develop an allosteric inhibitor. And our compound is very potent against PD4D. We know PD4D is important for normal brain function because ultra-rare neurodevelopmental mutations impact cognition in humans. So there are 35 kids worldwide that have 23 different missense mutations in PD4B, and these missense mutations actually destabilize the regulatory domains that are the target for the drug. So there's very good validation of this pathway, both through model organisms, through a terrific amount of academic research using earlier compounds, and now even human genetics. So this is the first PD4D inhibitor to enter human clinical trials. And the important feature of this compound's pharmacology is that it's a use-dependent inhibitor of the enzyme. So this enzyme is modulated by phosphorylation through protein kinase A, which is the key effector of cyclic AMP signaling. And so at the synapse, when cyclic AMP signaling is occurring, this enzyme is being activated, and then it ring-fences that signal. So it controls the temporal and the spatial pattern of information at the synapse. What the drug does is it doesn't inhibit very well the basal state of the enzyme. But when that enzyme is triggered and it opens up, then the drug is able to bind. And now it augments the normal pattern of spatial and temporal signaling for cyclic AMP. So that's a really important feature of the drug. And it's very similar to some of the most widely prescribed drugs that are treating CNS indications. For example, donepezil. Donepezil inhibits an enzyme that breaks down acetylcholine. What it does is it augments the acetylcholine that's being released in the brain in a specific temporal and spatial patterning that's important for learning and memory. The problem with donepezil is that once the cholinergic neurons disappear, there's no way of replacing the acetylcholine that's no longer there. In major depression, one of the most effective classes of drugs are the serotonin reuptake inhibitors. Again, these are augmenting the normal pattern of signaling by serotonin in the brain. So we're very excited by this profile, and it's a very unique pharmacological profile for a CNS active drug. So in parallel to developing the drug, we also had the challenge of showing where is the enzyme in the human brain, and whether or not the drug, BPN-14770, engages that target in humans. And so in parallel to developing the drug, also through a public-private partnership, this time with the National Institute of Mental Health, we're able to develop a PD4D PET tracer. So this is for the first time that anyone has been able to visualize in a living primate, in this case a cynomolgus monkey, where is this enzyme? And this particular PET ligand is sensitive to the activation state of the enzyme. So for the first time, we're able to see where this enzyme is in the brain. And it's very exciting because it's in the parts of the brain that are important for cognition. The labeling, the specific labeling by the PET tracer is shown on red on that slide. You're all familiar now with looking at amyloid PET tracer studies or tau PET tracer studies. So the same way, red indicates where the tracer is binding. And you can see it's in the prefrontal cortex. This is the front part of the brain. Here we go. Yes, okay. So here's the prefrontal cortex. It's present in the temporal cortex, entorhinal cortex, and also the hippocampus. These are the parts of the brain that are affected by Alzheimer's disease. These are the parts of the brain that are needed for cognition, for associative memory, for executive function, particularly in the front part of the brain. And what this also shows is that when we pre-dose the animal with the drug, we block the binding of the PET tracer. We now have first-in-human data in 2 subjects at the NIMH. We see the same effect in humans. The drug does enter the brain, does engage the target, and that the distribution of the target in the human brain is what we predicted from the studies in monkeys. So we're developing BPN-14770 for 2 different indications, for Fragile X syndrome and also for early Alzheimer's disease. In Fragile X syndromes, the connection between neurons and cortex and hippocampus don't mature properly. In Alzheimer's disease, those connections are lost. So the earliest clinical finding in Fragile X syndrome, the earliest biochemical change that was seen, was a change in cyclic AMP. Cyclic AMP levels are decreased. When the gene was cloned, it was possible to make both a Drosophila model of Fragile X syndrome as well as a mouse model of Fragile X syndrome. In both the flies and the mice, cyclic AMP is decreased. And in both those models, either genetically or pharmacologically, when you increase cyclic AMP, you improve you reverse the phenotype. So we, in collaboration with the Fraxler Research Foundation, were able to show that BPN-14770 increases cyclic AMP, it improves the morphology of the synapses, and it results in a global improvement in behavior in the Fragile X mice. So we're halfway through a 30-patient trial in Fragile X syndrome, and BPN-14770 has orphan drug designation in that indication. In Alzheimer's disease, the issue is that the connections are lost. So what BPN-14770 does is it triggers a compensatory mechanism. It triggers phosphorylation of CREB, which is important both for formation of short-term memories through modulation of long-term potentiation at excitatory synapses. But more importantly, it's important for stabilizing synapses. It's important for increasing brain-derived neurotrophic factor. And this pathway also modulates neurogenesis. It modulates the generation of new neurons in parts of the brain that are affected by Alzheimer's disease. And so there's a very interesting emerging literature now on amyloid and tau in human brains. And that's based on the use of these PET tracers. And what the PET tracers are showing is that at 80 years of age, 20 to 30% of cognitively normal elderly people have amyloid and tau in their brain. So some people are more resistant to developing dementia than are others. That's led to the concept of the development of the concept of cognitive resilience. Well, what does cognitive resilience mean? It means that there are compensatory mechanisms in your brain that can overcome the damage that's being caused by disease. And so we think this pathway may be that key to that compensatory mechanism. And so in animal models, it is possible to show that BPN 14770 protects from neuronal damage. This is an injection model where healthy mice are injected with A-beta 1 to 42 oligomers that causes loss of dendrites and spines. And then we see neuroprotection with BPN 14770. And this is also seen in human postmortem samples. So here's a series from a paper by Boros that was published in 2017, looking directly at those spines where excitatory synapses are being made and flipped in. Here's an elderly person, cognitively normal, no Alzheimer's pathology. Here's an elderly person, cognitively normal, with Alzheimer's pathology. So both BRAC positive for amyloid and tau. And here's an Alzheimer's patient. And what you can see is that the cognitively normal individual is somehow able to protect the spines on those excitatory synapses. So this is a very exciting mechanism of action. We're currently just launched a phase 2 trial in early Alzheimer's disease. It will be conducted across the US at 50 different sites. Planned enrollment is 255 subjects. In the first month, we've so far enrolled 10. This trial will report out towards the end of next year, and we finally have the chance of doing the experiment to see whether or not there's immediate symptomatic improvement in Alzheimer's. And I think this mechanism has the promise to both be disease modifying as well as perhaps to delay onset. So thank you very much for your time today. Tetra is built on a public-private partnership model, and I really wish the best for InvestiCure because I think there is a need for additional funding specifically for clinical trials. So thank you very much for your time today.